the organizers for inviting me. Second, when I just arrived, I got a little bit mystified because I was given a name tag identifying me as somebody from University of Notre Dame. First, I thought my university has been renamed, so I Googled it. No, it's still the same. Then I realized that I was undercover. But still, I just want you to know my real name and actually my affiliation, which is University of Notre Dame, which is pretty close to Chicago, 90 miles from Chicago. Usually people expect me to speak French, but I don't. All right, I'm going to talk about something completely different, as Monty Python used to say. I'm going to talk about blood clot formation. My interest in this conference is based on the recent kind of new collaboration with a group from UC San Diego. We're going to study Kawasaki disease, which manifests itself in formation of aneurysm in very small kids. And actually, as a result of it, blood clots are formed, and this results in death. So a lot of people are having blood clots for a variety of reasons nowadays. Maybe 20, 30 years ago, mostly older people and mostly people having like weight problem, people with diabetes. Nowadays, young people just have them, and if they're formed and they're not controlled, they get detached, they travel, and actually end up in lungs, and people die immediately, or go to brain, and people get paralyzed. People are getting very small blood clots formed during heart attacks. That's one of the main dangers now, because the heart can be jump-started, right? But then, as a result of kind of missing a bit, small clots are formed, and they go to the brain immediately, and people get paralyzed. Each year, there are, let me try it. There are at least 900,000 cases in the US alone, and then 300,000 people actually die of blood clots. Blood clots actually also result from heart surgeries. So I'll try to show an experimental movie. This is a collaboration with a group from medical school at Indiana University. And let me try. Well, let me explain what it is. I'll try to run it again. So they're using genetic markers. They're actually looking at small blood vessel in ear of a mice. They're shooting laser somewhere here. So they're producing injury, which starts internal bleeding. That's the main danger. So the blood is getting into tissue. This triggers very complicated cascade of biochemical reactions in blood, coagulation cascade. And smaller blood cells called platelets get activated, and they produce fibrinogen, and then become very adhesive. They stick together. So actually, in this particular movie, you can see them in green. So they're fluorescing in green. They stick together, form clusters. Actually, they form clusters which cover the injury, which prevents internal bleeding immediately. And then the whole clot grows, and the healing starts. And as soon as this happens, certain signaling occurs among platelets, and it starts disintegration of the clot. What happens in a lot of individuals nowadays, clots to grow to be too large and actually do not disintegrate. They either clog the artery, or they actually embolize. Embolization means they become irregular. The shape is very irregular, and big pieces of clot get detached, and as I said, travel along blood vessels, and people die. So let me just kind of indicate all the different components. Maybe could we switch it off? The lights. Right, thank you. So this is a cartoon produced by Ken Mann's lab from University of Virginia. This is the biological model, not computation biological. It's a cartoon. It has all the different components, and people explained what happens, and that's the model. Right? You don't need to run any simulations and feed parameters or anything. Okay, so you can see large blood cells. You've seen platelets. I'm going to run it again, if I'm lucky. All right, and then you'll see cut. Can be inflammation or cut in the vessel wall. Some chemicals are produced, diffuse into bloodstream, And then, here you go, starts bleeding, right? And then platelets become very adhesive, stick together, produce fibrinogen, the blue stuff, and cover the injury. Looks really exciting, but it's a cartoon, so it's a disclaimer. It's not simulation. So what we've done, we used confocal microscopy, and we looked at time sequence of a growing blood clot in specific animal. So our collaborators are producing genetically modified mice with different levels of factor 7. So they actually calculate in a different way. So one rotation corresponds to one snapshot, three-dimensional reconstruction, actually, of the internal structure of the blood clot. Green stands for actually a fibril network, and red, in this particular movie or sequence of images, actually denotes activated platelets. So I mentioned fibrinogen. The whole setup is really complicated. So platelets, when get activated, they adhere to each other and then produce fibrinogen, which helps, actually produce thrombin, I'm sorry, which affects fibrinogen in blood, and it starts polymerizing into fibrin, and then into fibril network. So initially, blood clot is like a jello. It consists of mostly activated platelets. And actually, if you look at this carefully, you see it in this experimental sequence. And then this fibril network actually cements the clot. So later, it becomes pretty elastic, but still it can withstand considerable pressure coming from the blood flow. Well, we set up a multi-scale model consisting of different submodels. Our focus is on kind of matching time and space scales, and we're using different approaches for this. So the model consists of different types of cells carried in blood. So there have been a lot of very interesting papers by a group of Aaron Fogelson from University of Utah, and of course they're using immersed boundary method for that. We're using something else, and I'll explain this in a second. But basically, we model different types of cells moving in the plasma, in the flow. And then there's an injury site. There are certain chemicals which diffuse into the bloodstream. And we're using incompressible Navier-Stokes PDE solver for the blood. So these are all the basic components. And these are different scales. So we look at the subcellular level. So we're modeling coagulation, anticoagulation pathways in detail. Because when clot grows to be the right size in healthy individuals, it starts disintegrating. So in many cases, it's not what's happening now. So we're trying to understand what might be the reason for that. Okay, then next level is cellular level. We're using cellular POTS model for that, which comes from statistical mechanics. I'll try to explain it if I have enough time. And then the highest possible level, it's the level of the blood flow, Navier-Stokes, and advection diffusion equation for chemicals. So we have different grids, which we superimpose. And there is a fixed grid for cellular POTS model. And then we have a grid for PDE solver. And of course, we have an interface, and it's a growing interface. I just want you to understand the complexity. So platelets actually adhere to the surface of the clot non-stop, so that the geometry changes all the time and affects the flow. So platelets actually go through different stages. They change their states. Let me talk about cell representation. So it comes from the Ising model from statistical mechanics, where it was used for modeling magnetic materials, just phase transitions and so on. So basically, we have a lattice, can be three-dimensional lattice of different topology. And then each cell is represented by a cluster of pixels. And it's a Monte Carlo, it's a stochastic system. We're using Monte Carlo simulation. We flip specific point, it can change its type. So it's cell, type 3, number 17, whatever. We look at the boundary and we flip it, and we recalculate the Hamiltonian, the energy of the system. And the different parts of the Hamiltonian, basically it's a pixel by pixel interaction, and we can actually model cell-cell adhesion in detail using this particular approach. But then we have kind of specific additional terms just to make it more kind of biological. So in this case, we're looking at clusters. We don't want them to shrink or to grow to be too large. So we have target volume, target area, and we have a efficiency function. So if you flip too many times and it grows to be too large, the probability of this happening again is actually going to be reduced. So if we flip specific pixel, change the type, we calculate the energy. If it reduces the energy, we accept it with probability 1. If not, we calculate the Gibbs measure, and whatever happens, happens. We use generator of random numbers. But outcomes with higher probability, if we do it many, many times, millions of times, actually happen with higher probability in the end. We embed our cells, clusters of pixels, represented on a different grid, into the flow. And we switch from one calculation to another, once so often, matching time scales. And we incorporate additional term in the Hamiltonian to account for the shear pressure coming from the flow. And this is how we calculate it, just integrate along the boundary. And basically, it encourages cells to add more pixels on one end and lose them on the other, and move in a specific direction. So this is our simulation. And you can see basic interesting things happening here. So this is the injury site. You don't see the continuous part. I'll show it in a second. And then these activating chemicals are produced. Platelets get activated, adhere to each other, cover the injury, start growing. And then something else is happening, actually. When it grows to be specific size, you see that there's some turbulence forming here. And a lot of larger blood cells, which are not that adhesive at all, get pushed inside of the blood clot. And if I were to have some time, I would go back and show you the initial simulation, and you'll see that internal structure of a clot is extremely inhomogeneous, and it contains a lot of larger blood cells. It undermines the stability of the clot. That's the real danger. That's one outcome. And also you see actual formation of thrombi, and we played with different parameters, and we can get different outcomes here. So this is the continuous part of our simulation, the turbulence, the vortex actually forming behind the clot. So the clot is growing somewhere here. With certain Reynolds numbers, we kind of tabulate it carefully. We get this kind of phenomena. We calibrated the model. So we ran simulation where we looked at dependence on the size of the clot for a certain period of time, on the flow velocity, and this is the simulation result. And we got pretty good match with the experimental result. Then we looked at pulsating flow, and then looked at non-Newtonian case, very small blood vessel. This is kind of preliminary, but still we see the trend, which looks very promising. We're also analyzing very carefully the flow close to the clots with a kind of complicated geometry, and we used the Lagrangian coherent structure approach developed at Caltech to separate different parts of the flow. Because, for example, in this case, this particular configuration actually captures platelets, and the clot can grow this way very rapidly. And in experimental images, we see this kind of structure which consists of many, many different peaks all the time. All right, so I never talked about elastic properties of the clot. So we're currently coupling our cellular POTS model with the so-called subcellular element modeling approach, which incorporates cytoskeleton, basically a system of nodes which are actually linked to a system of springs. And it's more along the lines of molecular dynamics simulations. But I think now we're capable of kind of matching those different models together. It's more important, actually, it's important to use this kind of approach if one wants to parallelize the code. And this is the focus of our research as well, because we would like to run simulations for millions of cells. And in this molecular dynamics approach, we'll be using Leonard-Jones potential and elastic potentials. Okay, so the one prediction which we made already using our model is the importance of factor 7, which kind of controls the anticoagulation pathway. So this is the first kind of available data on the structure of growing clots in mice with different levels of factor 7. So the coagulation levels are different in those mice. And in normal wild-type mice, actually, it grows to be specific size, and then it stabilizes. And we see formation of fibril cap, which kind of covers the whole clot and actually interferes with activation of platelets. And this is the case of low level factor 7. So we added to our model, and this is how they look actually. The clots which we obtained in those two cases look very different, and the sizes are different. So we kind of extended the Kahneman's model and biochemical reactions, and realized that in case of a mice with limited factor 7 production, actually, production of thrombin is delayed, which interferes with the final size of the clot. So this kind of effect interferes with early activation of platelets. If they don't get activated, they don't adhere, and the clot starts growing later, and then it kind of affects the final outcome. All right, so we're validating model. We're close to implementing first 3D kind of implementation for this particular setup. And we're also looking in detail at the structure fibril network. There's a lot of literature on modeling gels. This is gel which is full of cells, and these cells are controlling the growth and then disintegration of fibril network, which makes it more complicated, more interesting. Well, thank you for your time.
Thank you very much. Maybe one short question?
Maybe I missed it, but the fibrinolysis rates, were they factored into the rate constants for those were factored into your Monte Carlo simulations?
Yes, so through biochemical reactions. So we're modeling actually production of thrombin on the surface of each individual platelet.
During clot formation, those rates are probably changing.
That's right.
And it's very, very difficult to really pin down the experimental values for that. Are you able to do that?
Not us, but there is a group of Diamond at University of Pennsylvania. That's exactly what.
Yeah, it's my colleague. It's in my
institute.
institute. That's why I asked.
So that's the next step, actually.
Yeah. Well, thank you. I think all the presentations